So today's speaker, Pete Warzybok, is a senior marine ecologist and Farallon program leader. He focuses on ecology and conservation of marine ecosystems, as you may have guessed. He oversees activities at our long-term research station on the southeast Farallon Island and maintains our long-term monitoring data on populations, breeding success, the timing of things, also known as phenology, and diet of marine birds and mammals. Pete is a New Yorker. Well, he grew up in New York and earned his Bachelor's of Science degree in biology from the State University of New York at Purchase. And Pete joined the Farallon Islands program as a volunteer seabird research assistant in the spring of 2000 and was hired as a program biologist the following year. And then after several years working on the islands, he returned to grad school at Humboldt State, where he studied common murre ecology, one of the most numerous birds on the Farallon Islands. And to date, he's spent over 1,700 days. Pete, do you want to correct that stat?
A little over 2,400 at this point.
Okay, we got to update your bio. Nice. On the island. So that's pretty impressive. And when he's not on the Farallons, he's now working out of his home office in Minnesota, like you saw in the chat earlier, working on data management, analysis, manuscript preparation, fundraising, outreach, and logistics support for Farallon staff. His current research interests include diet foraging ecology and ecology variability in relation to breeding success and population dynamics on the island, on the Farallon seabirds. So thank you so much for sharing your work today, Pete. And before I pass you the mic, I'm going to remind everybody again to jot down your questions as Pete shares, and I'll be doing the same with you. So with that, I'll pass it to you, Pete.
All right, great. Well, thank you, Lishka, very much for that. Let me see, I think this one. Do you see my presentation now? Great. All right. So again, thank you, Lishka, for the introduction. Thank you, everybody, for joining me here. As the last speaker in our marine series, I'm going to do a little bit more of an overview on how we use our seabird diet data from the Farallons in various conservation projects. So obviously, Farallons is a long-term project. I'm the one talking today, but it is really a group effort, the whole California Current group and the many research assistants and biologists that came before me collecting some of this great long-term data. There we go. So seabirds as fishers. There's a couple of examples of our Farallon seabirds going out and doing some fishing for us. So seabirds, by definition, are those species that obtain their food primarily from the sea. And most of that food comes in the form of these small, what are called forage fish. So things like anchovies and sardines that you might be familiar with. And seabirds are extremely good, extremely efficient at finding and obtaining food from this vast area of the seemingly uniform ocean. And that makes them really great environmental samplers. And they are really good at indicating what's happening in the marine environment, telling us about the abundance and distribution of these forage fish prey in the environment. And then we can take some of this data as well, and we can relate it back to how to breeding success of the seabird. So what their diet, how that relates to breeding success, to changes in their populations, to environmental conditions, warmer water conditions, El Nino events, things like that. But we can also learn a lot about the forage fish themselves. The seabirds are really telling us what's happening in that environment, and they're tracking the populations of these forage species for us. And so when we're talking about gone fishing Farallon style, it's a little bit different than what you might think of as going fishing. We don't take a pole and go stand on the shoreline to catch fish or throw a net in the ocean. We wait for the birds to bring the data back to us. And there's a couple of different ways we do this. One of them is diet watches. So for common murres and pigeon guillemots, they bring back single prey items to feed their chicks during the summer. And we can sit by the colony with binoculars and identify those fish as they're flying in to the colony. So that's what we're doing in that photo on the upper left of the screen. We're doing a diet watch for pigeon guillemots down at Garbage Gulch. And you might notice that 2 of us are actually doing diet watch and 3 people are looking at the camera, but usually we're all on point, focus on the birds and the fish. So that's one way that we collect fishing data. Another way is actually by collecting fish from the birds themselves. So in that lower image on the left, that's Katie Saunders. She was a RA back in 2011, and she's removing a rhinoceros auklet from a mist net. So we have a long-term mark recapture study with rhinoceros auklets, where we're catching them and banding them and looking at the return rates and survival. But while we're doing that, they're coming in with a bill load of fish. So in that first slide I had, you saw the auklet in the middle had 8 or 10 fish in their bill. So they'll carry a bunch back at a time. And when they hit the net, they drop the fish on the ground and we simply go over and pick them up. Then we bring them back to the lab and somebody goes through and identifies them to species and takes some morphometric measurements, length, body depth, and we take a weight on them. And so that's what Mike Johns is doing there in the upper right corner. I believe he's measuring a small salmonid and collecting data on them. And then finally, for some species where the fish are not visible, like Brandt's cormorants, they will regurgitate a pellet just like an owl does. And this pellet is like a little ball of kind of disgusting mucus full of all the parts of the fish that they can't digest. And so we go out at the end of the season out into the colony and we collect up these pellets and we bring them back to the mainland to the lab there in Petaluma. And Meredith and the wonderful folks in the lab crew are the ones who actually go through these pellets and they pick out the, fish otoliths with ear bones, scales, squid beaks, vertebrae, whatever they can to help identify the prey items that the cormorants are bringing back. So not traditional, but some unique ways of going fishing. And these are the kind of regular culprits that we find in the diet. The northern anchovies and juvenile rockfishes of a bunch of different species are the most common items in the diet. We also get juvenile salmon that are migrating out to the ocean for the first time from their inland river spawning areas. So they come out of the bay and our seabirds will take them as they come out. We get the oceanic types of smelt, so not necessarily the endangered delta smelt or bay smelt, but the other species that are more common out in the open ocean. We also get some invertebrates like market squid and krill, and several types of flatfish, including halibut and sand dabs. And again, that's the juvenile stages of those. And then this is what the long-term data looks like. And I know there's a lot going on, having 4 slides on one figure or 4 graphs on one figure, and you don't really have to pay too much attention to it. It's just to demonstrate the long-term time series that we have for diet data from the Farallons. And I chose 4 examples, 4 different species. There's a few things that I just like to point out. So the 4 species that we have here, common murres, rhinoceros auklets, pigeon guillemots, and Brandt's cormorants, all sample kind of different parts of the marine environment. So we're getting a very complete picture of what's happening around the island. Murres are deep divers. They'll dive to over 100 meters, but they feed primarily on the shelf, on the waters above the shelf. So they're diving from the surface all the way to the bottom, and they target primarily fish that are up in the water column. And you can see from the colors on there that the dark blue and the yellow are the most important. That's anchovies in blue and rockfishes in yellow. Rhinoceros auklets, also a diving seabird. They don't dive quite as deep, maybe 60 meters, but they tend to feed outside the island. So at the edge of the continental shelf break and beyond at these submarine canyons. So you can see a little bit more variety in their diet composition than you do with the murres. Pigeon guillemots are extremely near shore and they feed in the benthic environment. So they're feeding really close to the island. They're going down to the bottom. They're taking things from little rocky crevices and reefs around the island or from the sandy bottom areas. And so you see, in addition to those rockfish, you have the pale blue, which are the flatfishes, and then the turquoise, which are sculpins, are all showing up in the guillemot diet. And finally, you got the Brandt's cormorants that are, puking up those little pellets for us. They actually go more towards the mainland. So they're feeding on the shelf, but they're going coastal, and they're feeding on pelagic schooling fishes near the coast. So we're getting this wonderful long time series covering the entire ecosystem around the Farallons. So for the rest of my talk, I'm going to focus on 3 different ways that we use this long-term data. And the first one is we like to make sure that we're using this data to make sure that the wildlife needs, so their food needs, are included in fisheries management. So this is just a little figure showing what a stock assessment model looks like. Essentially, fisheries management plans and harvest quotas, so the amount of fish that you're allowed to harvest, are typically based on these stock assessments. And it's basically an estimate of fish population, and it's based on estimates of the fecundity, recruitment, spawning biomass, age and maturity, and different sources of mortality for each species of forage fish that you're trying to estimate. And the 2 things that I'd like to point out on here are the 2 sources of mortality. So fishing mortality, that's how many are being captured by the commercial fisheries, and that's really easy to measure. We know how much is being captured. The other is this natural mortality component on the lower right, and that's where our seabird diet data really comes in. With that seabird diet information, we can make some pretty good estimates of how many fish are being consumed by predators in the marine environment. And then that gets factored into these models so that they can set more realistic harvest limits for these forage species that are important to seabirds. And the way we estimate the natural mortality for these fish species is by building what's called a bioenergetics model. Many of you might be familiar with these from different environments. They're a pretty common tool. It's mostly just an additive model. And what it does is it estimates how much food is required to meet the daily energy needs of a particular predator. So the different factors that go into it. So we're trying to figure out, say, how much food a murre needs in a day to maintain itself. So there's a few different factors that go into this. One is the actual energy requirements for that bird. And that's based on studies of their field metabolic rate or their basal metabolic rate adjusted for activities. So we know how much energy they need to maintain themselves. Then we have our long-term diet time series, which tells us about the fraction of the food item in the diet, so which different prey species they're selecting in any given year. And that might be a function of environmental conditions. For example, El Niños, warmer water conditions, you get more anchovies. La Niñas, colder water conditions, you tend to get more rockfish. And this is important for the next factor, which is the energy density of prey. So each fish is worth a certain amount of calories or kilojoules of energy, and that varies between species. Anchovies, for example, have a much higher lipid content. They're fattier fish, so they're worth about 5.6 kilojoules per gram, where a juvenile rockfish is worth only about 4. So depending on what they're eating, they need to eat more or less to meet those daily energy needs. Then finally, there's the assimilation efficiency. So how good is a murre at converting the energy from the fish into energy it can use? And so we roll all this stuff up into the bioenergetics model, do it for daily, multiply that out for a year, and then multiply it by the population to get this estimate of how much food they need. And these are some of the variables that go into these models. So obviously the seabird diet composition I've already talked about. Population, if we're trying to get an idea of the total consumption so they can set reasonable harvest quotas, we need to know how many birds are out there consuming it and other predators like seals and sea lions, other predatory fish, in fact. And then climate plays a factor and environmental conditions. So we know that sea surface temperature and upwelling conditions have a large effect on the prey resources available, what these forage fish are doing, how many there are, where they're concentrated. And then larger scale, those initials there at the bottom are larger scale environmental indices that kind of tell us overall conditions in the Pacific Basin. So it can wrap all of that stuff up into the bioenergetics model and get a reasonable assumption or a reasonable estimate of how much food that these birds are going to need. And this is an example of what it looks like. So this is from a paper we did back in 2018. And this was very limited. This was just for 3 species of seabirds from the Farallons during the breeding season. So very limited spatial scope to this. But even with that, we found that the amount of fish they need to survive in a given year ranges from as little as 8,500 metric tons to greater than 60,000 metric tons over the course of time. And if I had time to add the last few years to this time series, we would have seen that go up even more as the populations continue to grow. So a couple of things that we take away from this: the amount of prey that they're consuming by species is obviously pretty variable. Some of those years, the blue, the anchovy is much more prevalent. Some of it, they're getting most of their energy from the rockfish. And also, as these seabirds have recovered from past disturbance and these populations are growing at the Farallons and in the Central California region, we're seeing them taking a much greater proportion of the forage fish that are out there. And this is important for one big reason, is that harvest limits really haven't kept up with changing environmental conditions or seabird populations. So this line that I just had pop up on there, that's the anchovy harvest limit, about 25,000 metric tons. And that was set after a stock assessment in 1995, and then hadn't changed until just this past year. So for more than 20 years, they were sticking with that 25,000 metric tons that you could harvest of anchovy in this region, but they were not factoring in the huge increase in consumption by seabirds over this time. So in the early years, it was maybe seabirds were consuming less than half of the allowed fisheries take. Now they're consuming more than double the allowed fisheries take. So being able to factor in predator needs is really important for making sure that we keep these fisheries sustainable. And what we want to do from here is scale up. So I mentioned that was a very small spatial scale that we looked at that first time, but now we want to expand to the entire West Coast in the California current ecosystem. And so this is just a kind of a straw man idea here. This is a mock-up that we did for a proposal. If you were to look at common MERS, for example, let's assume a ratio of 60% anchovy, 20% rockfish, and 20% other. And then we looked at how many MERS are in these colonies along the coast. So that figure on the right shows all the major MER breeding areas with estimates of their populations. So if we take each of those and we look at that static estimate of the prey proportions, then we come up with these estimates of prey consumption that are represented by the bubbles. Of course, we would want to use real data for this. This was just a mock-up, like I said, and we can get population estimates for all of these colonies, and we can get at least regionally specific diet information to roll into this model. So Mike has been leading this work for the last couple of years, and we're hoping to roll this forward over the next year or so. And then we're also talking about moving beyond the California current. We are the California current group, but we are also all about increasing pace and scale of climate-smart conservation. So we're trying to get beyond. And one intriguing area of interest is the Humboldt Current off of Peru, where there is one of the largest fisheries in the world. The Peruvian anchoveta fishery is among the largest. More than 10 million tons of anchovies are harvested there each year by the fisheries. It's also critical prey for very large populations of marine birds and mammals. Many of you might have heard of like the Guano Islands off the coast of Peru, where these big seabird colonies. And so they're also dependent on these anchovies. But 75% of the anchovy stock is estimated to be taken by the fisheries, leaving only 25% for all those other predators, and also to maintain the anchovy population. So unfortunately, in their traditional fisheries models, they have not considered predator needs, but they want to start doing that. So we submitted a proposal to the Schmidt Marine Technology Partners a couple of months ago, and we're partnering with some researchers in Peru that Jaime knew from his early career. And we're looking at ways that we can employ these kind of bioenergetic models to the Peruvian fishery to help it set more realistic harvest quotas there as well. So the first part there was talking about how we can improve fisheries models using our data and make sure that the predators are represented in that. The second way we're using these data is to actually look at trends in the fish populations themselves using the seabird diet. So traditionally, fish stock assessments and population estimates are based on trawl data. So people go out in a vessel, they tow a net, they collect fish, they count them, and they evaluate trends over time. But there are some constraints to doing trawl data. They tend to be limited. You can't do them very often. They're very expensive. You can only do them when the weather is good. You can probably only do them seasonally. And other things like vessel size make an impact because larger vessels can't survey near shore. And finally, you're surveying, by design, you're surveying at set transect points. So in the figure on the left, that's a map of the access cruise, Point Blue's access cruises, showing the different transect lines. And the triangles are where they're doing the trawls. But fish populations, these concentrations of forage fish, are not evenly distributed across the environment. So you may or may not get a representative sample by surveying only at these locations. Whereas seabirds are surveying everywhere. They're going to find the food wherever it is. So they're getting, and especially when we combine those 4 species, we're getting a much more complete picture of the forage fish around this region. The 2 figures on the right are just to show you some changes based on the trawl data in anchovy and rockfish relative abundance. So I won't get into that data. It was just already on the map. One other challenge with vessel survey data is that you can't always do it. So not only does weather affect it, but something like a global pandemic can shut down NOAA fisheries for a couple of years. And that's what happened in 2020. They had to cancel both NOAA, the National Oceanographic and Atmospheric Administration. They do surveys for fish stock assessments every year. They had to cancel all their trips, and our access cruises had to be canceled in 2020. And then in 2021, several of the NOAA cruises were either canceled or reduced as well, leaving them with these big gaps in their time series. So once again, seabirds come to the rescue. Seabirds are really good at tracking fish stocks. And as we know, they're sampling everywhere. So what we did is we partnered with some researchers at NOAA, Jared Santora and others, and we looked at how well seabirds are tracking trends in the abundance as indicated in the trawl surveys. So basically what we're looking at is, does the data from our seabirds tell us the same thing as the data from the trawl surveys? And we published that in this paper about a year ago. And so the important things to look at on this panel of figures are the top 2 figures show the relationship between, in this case, common murre diet and the trawl survey data. And you can see on the left is rockfish, on the right are anchovies. And you can see that trend line and that really strong relationship between the survey data and our diet observations from the Farallons. So that was really good. We were hoping to find that. And so then what we did next is we took that information, that relationship, and we said, okay, if you can't do a survey, can you replace that survey data point by using the diet data from the Farallons? And so we created a model. I should say, Jared created the model. He's a really good modeler, but they created a model that used the seabird diet data to predict what the trawl survey data would show. And we used the time series going back several years to train that model. And then we predicted what the trawls would catch. And that's what the bottom 2 figures show. So the blue line is the actual trawl survey data for all the years where it was available. And the red line is the model predicted based on seabird diet. And so you can see that they track each other really well. And so the seabirds do a very good job of predicting the data points in the trawl data, the fish abundance as related by the trawl data. And the places where it doesn't line up may be a factor of that sampling error from the trawl surveys that I talked about. Again, there's places where you may get overrepresentation or underrepresentation in those trawl survey points, but the seabirds are sampling everything uniformly, so you get a little more even coverage. So finally, the third way that we are using our seabird diet data, our forage fish data from the Farallons, is to help guide management and ensure sustainable fisheries. One of the things that we do is we share this data openly with management agencies. And we contribute this data to these large ecosystem reports so that anybody can use them. Two of these are examples right here. The State of the California Current Report, which is produced every year by a group called CalCAFI, stands for California Cooperative Oceanic Fisheries Investigation. It's quite a mouthful, but they've been studying fish populations for decades, and they have determined that our diet data from the Farallons greatly improves their ability to track fish trends. So they're including it in their annual assessment report. And the same is true with the National Marine Fisheries Service, a division of NOAA, where they have an annual report on the state of the California current. They use a suite of environmental or ecosystem indicators, among which are the seabird diet data from the Farallon. So it's a critical ecosystem indicator for the state of the California current. And then just recently, we had a couple of really important impacts on management. So we have been engaging with the Pacific Fisheries Management Council, which is the governing body for all commercial fishing activity on the West Coast. And there were two big things that Farallon Data contributed to in the last year. The first was with northern anchovy. So as I mentioned earlier, there's been a static catch limit of 25,000 tons for 20 plus years without any change to consider with increasing predator populations. Well, at their meeting in January of this year, after several meetings with us and reading that paper and talking with other experts in the field at NOAA, they have revised how they set harvest rules going forward. And now there will be annual evaluations to set annual catch limits that will be based not just on stock size, but also on the needs of predators as based on those bioenergetics models that I talked about earlier. And then the other big accomplishment from a management perspective was preventing the development of a new fishery that would impact forage stocks. So we demonstrated with our data, again, how important juvenile rockfish are as forage for seabirds and other predators. Those yellow bars and all of those figures I was showing, those are all rockfish. And there was a petition to start a new directed fishery for juvenile or for short-bellied rockfish, which had never existed before. And so we met with the management council, we presented our data, we wrote letters expressing our expert opinion, and as did other cooperators, Audubon, Nature Conservancy, the National Marine Fisheries Service, and the Fisheries Management Council decided to decline that petition and protect the stocks of short-bellied rockfish to make sure that they're available for predators like seabirds and marine mammals. So that's what I have for you for today, a few of the different ways that we use the fish data from the Farallons. Thank you all for listening, and I think we got time for questions. I actually have no idea how long I talked.
Yay, you can put your video on and give Pete some love with your clappies or there's lots of emoji clappies happening too. Thank you, Pete. That was awesome. Yeah, we've got 20 minutes for a Q&A. So yeah, go ahead and use that hand raise function in the reaction section or the chat box to ask questions. And I just want to say, like, as I was listening to the talk, I was like, wow, this is like a really great example of including the audience in solving a problem that's affecting the audience, which is seabirds. Like, you've detected an issue and you're kind of like asking them like, what's going on? And then they're part of the solution. So it's pretty cool.
They're telling us what's happening out there.
Yeah. Yeah. All right. Looking for the hand raises. I don't see any yet. Oh, let's see. I see a thing. Okay.
I see a question from Julian in the chat.
Yeah.
Asking how confident we are in IDing the prey items in the bills of the birds. Pretty high confidence. A lot of them are fairly easy to distinguish from each other, although sometimes we have to group them together. So just like identifying a bird that's flying by, there's specific field marks you look for, coloration, shape and pattern of the fins in relation to each other, size of the prey item. And so you can use those after a bit of training to easily recognize what prey. Now, there is a limit to that. So there's, I think, 14 or so species of juvenile rockfish that we've collected in the rhinoceros auklet diet. We can't tell them apart in the visual ID. We can just classify them as rockfish, but we can't tell which species.
Nice. Well, our participants are, oh, is it going to recognize your hand visually? No.
Didn't work. I tried.
Okay, I see your visual hand, Mike. Go ahead.
So Pete, in all of your many years out there on Cepheid, what is the biggest, the most surprising diet-related thing you've seen in any of the species?
Ooh.
Trend or appearance?
I mean, that's a good question. Difficult to answer. There have been a lot. As you noted, I started out there in 2000, so it's been 22 seasons of seabird work out on the Farallons, and there's a lot that's gone on. But I think there have been some very strange fish that we've seen. Lamprey eels and the Medusa fish and things like that, just really strange prey items that show up. And this typically happens in poor years when their preferred prey, anchovies, rockfish, flatfish, are not available. Then you start to see this crazy variety of things show up in the diet, which is fun when you're doing a diet watch, but is not good, a good sign for the ecosystem.
Cool. And we've got a question in the chat from Dennis. Have you been able to say anything about the salmon stocks using this data, given it's a relatively small part of the diet? Good
question. Thanks, Dennis. I actually didn't want to drag on for too long, so I cut out a slide where I talk specifically about that. But yes, we did a paper with Brian Wells at NOAA a few years back, where we used the salmon data from our observations. And what we found is that even though it's a relatively small proportion of the diet, it does show, it correlates really well with trends in survival and recruitment of salmon. So when MERS, for example, are eating more salmon, even if it's not a huge volume of salmon that shows up in our diet watch, when it's higher than average, then you see lower survival, lower recruitment of the salmon when they're returning to spawn a couple of years later. And so it's a good indicator of that. And the other thing that was really cool from that paper is that prey switching, again, is really important. When MERS are feeding on rockfish, they're feeding out near the island and they're hardly ever getting any salmon. But when they're feeding on anchovy, the anchovy tend to be concentrated a little bit closer to shore, near the mouth of the bay, which is coincidentally where the juvenile salmon are coming out. So they end up taking a lot more salmon during an anchovy year than they do in other years. And that shows up in the salmon recruitment later on. So now the salmon researchers are also using Farallon diet data to improve their recruitment models for salmon populations in the Central Valley.
Sweet. Very cool. Good question, Dennis. And then we have a question from Aiden. At what spatial scales do the diets of seabirds you monitor vary? How wide of an area are the Farallon diet observations applicable over?
It's a good question. So they're applicable, essentially within the foraging range of breeding seabirds. So when they're breeding, they're tied to the colony. They're what's called central place foragers. So they have a kind of limited range around the island that they're going to go to get prey. For us, that's effectively, I don't think I could put a square mileage on it, but from about Bodega Bay down to, Año Nuevo, or maybe just a little bit south of that. And from the shoreline out to, a few miles past the continental shelf break, which is maybe about 35 or 40 miles offshore. So the diet from the Farallons, I think, is representing that area fairly well.
Cool. Okay, we've got a question from Julian in here. Have you seen any response to fish harvesting? Is there any correlation between excessive commercial harvest of a particular fish species and what the birds bring in?
So no, we haven't seen too much of that. So even with the anchovy, well, what happened was there was a big crash in the anchovy population, and the fishery was actually shut down for a couple of years to prevent just that thing from happening. But we haven't seen a strong correlation between harvest limits because it's been fairly low. We haven't seen a correlation with seabird productivity.
The harvest limits are fairly low.
Well, I mean, it's been set at 25,000 metric tons, but it hasn't been the last few years as this population is exploding. The actual amount harvested has been less than that because it hasn't been commercially viable to go out and harvest it to large degrees. So as the anchovy populations now are recovering, in fact, this past year seemed to be record highs for anchovies in this region. Now there's talk about ramping that fishery back up. So we need to make sure that it doesn't go too far in the other direction.
At one point in your talk, I think you mentioned something like alluding to the fact that the fisheries management folks, maybe that are part of the fishing community, they want to include predator needs in fishing management. So I guess I'm just curious if you can give a little bit more insight into like what the relationship is like with us as scientists and the industry, like, and how our science sort of affects business and consumer experience, that whole kind of like interaction.
Right. So what I was referring to was the management agencies, not necessarily the commercial fishers themselves.
Okay, that makes sense.
So we have very close partnerships with the NOAA National Marine Fisheries Service, the Pacific Fisheries Management Council, and Cal Fish and Wildlife, which manage the stocks at different scales. The commercial fishermen themselves, we haven't gotten a lot of feedback, at least Point Blue hasn't. I'm sure the Management Council has, because there's commercial fisheries representatives on the Management Council. But the fact that they chose to adopt those new regulations, even with representation from the commercial fisheries, indicates to me that they're at least willing to consider the needs of predators when setting harvest limits.
Cool. Yeah. Curious about like that whole end of the spectrum of like how I'm sure there's a diverse array of perspectives amongst fisher folks.
Right.
But
So far in our region, Gulf of the Farallons region, there hasn't been a lot of direct competition and interaction with the fishermen themselves. Now, if we do as we hope and expand along the coast or to Peru, that will probably be a lot more contentious because the fishery is so much so much more of a part of the economy there, and they're harvesting so many more fish that reductions or regulations on that for the needs of predators might be a lot more contentious than they are in this region.
right, I need to consider a partner biologist program of implementation down there, huh?
Mm-hmm.
Cool, thanks, Pete. Brad has a question. He's wondering if climate projections are factored into how these fish populations may be affected in the future.
Yes, if we were to project forward, so the example that I showed was using existing data to determine how much they're consuming on a year-to-year basis. But if we are to project forward, then environmental conditions and future climate predictions would also be factored into this model. So as you look at the frequency of warm water events like the blob or marine heat waves, we'd want to factor those in because that greatly impacts the forage fish community. And the frequency and the intensity with which events like that happen must be considered to be able to project the needs of predators going forward. So the answer is hasn't been to this point, but will be going forward if we're doing projections, which hopefully we get funded to do.
Nice. Okay, one more, maybe comment and question from Aiden. Could you use stable isotope analysis on feathers or other methods to determine non-breeding season diet?
Excellent question. Yes, to some degree you can. Stable isotope analysis has limitations. Largely, you can't distinguish between species using stable isotopes. You can more or less distinguish between trophic levels, unless you have specific ratios from those prey items. But even then, like the stable isotope ratios from, say, a rockfish versus an anchovy are going to be very, very similar. So it'd be difficult to determine species content, but you could do things on a broader scale in terms of how much krill versus other invertebrates like squid versus fish, that kind of thing you could get at. So that is, though, it is a good thing to note that that is one of the challenges with trying to scale this up to a year-round consumption estimate, is that the information on diet during the non-breeding season is much more spotty. There's not as much available and the time series are not as rich. So there's a challenge there.
Hand raised from Julian. Go ahead.
That reminds me of the use of what do they call it?
eDNA.
The environmental, like the genetics is just in the environment, in the water, basically.
Right.
To assess what's there. Is that even a possibility for determining like the fish population, I guess?
You know, it's been mentioned. I don't know enough about eDNA to know if it would work on that scale. It would certainly detect presence absence of different species in different areas using eDNA, and it's an amazing tool that's being developed, and they're using it in lots of different ways. So you could use it to determine if anchovies are present, but I don't know if you could get a relative abundance measure from them. You might be able to, I don't know.
That makes sense.
But it is an exciting field going forward and something we've discussed a little bit here.
Looks like Meredith chimed in about it too.
There you go. Apparently we're already starting to do it. And that's
Awesome.
Just another thing. We got to have all the things sampled on access cruises. So we got to make sure everything that we can think of is happening, and it is.
Thanks, Meredith.
Sweet. Well, thank you, everybody. That was really wonderful, Pete. Great talk, really informative, great questions. Thank you for wrapping up our oceans theme for 2022. And everybody, if you want to see Pete on the Farallons, if all goes well, you can join us next Wednesday at 11. And Pete will hopefully be broadcasting live from a fur seal colony. If technology is our friend, we'll find out soon. But either way, something about fur seals live from the Farallon somewhere next Wednesday. And next Thursday, we're going to kick the Shorelines talks off with, I think, Megan and Julian. Definitely Megan, talking about marshes. So I'm going to be sending out our schedule and calendar invites today or tomorrow for that. But you can go ahead and just mark your calendars. Most Thursdays, 12 to 1 Ecolunch. Gonna schedule some good stuff going throughout the rest of the year and into the next. Yep, and I think that'll wrap it up for us.
All right. Well, thanks everybody for joining us today and listening to me talk about fish. Maybe I'll see some of you at the office tomorrow.
Nice.
All right.
Bye.
Everybody.